Yeah. Okay. A very warm welcome to all of you to this session. So today we are going to discuss genomics, what is genomics data, how it is organized, and how it is interpreted to be such a powerful resource in the hands of clinicians, researchers, and entrepreneurs, and how reading the DNA code will help us battle cancer, will help us fight off infectious diseases, and will help us even solve some crimes. So let's start by reviewing the fundamentals of genomics. So genomics is all about DNA. So let's review what is DNA code. So what do we already know about this, and what kind of answers can we find here? How can we analyze huge sequences of nucleotides stored in the DNA, and how do we make sense of them? Just like a book, the DNA code is organized as letters that form sentences, and sentences that form paragraphs, and paragraphs that form that are organized into chapters. Knowing how to read this code can help us decipher the full story about who we are, and to look into our history and to understand how we are connected to other living things around us. So DNA is short for deoxyribonucleic acid. So it's a long molecule composed of two chains that coil around each other to form a double helix. This double helix carries genetic instruction for development, for function, for growth and reproduction of all known life. So this all known life includes multi and single cell organisms, and as well as many bacteria and even some viruses. So yeah. So the DNA code contains directions or recipes for life, including the capacity to grow, including the capacity to reproduce, and including functional activity that characterizes the cells, and including the continuous change that proceeds until the cell dies off. Since life is a condition that distinguishes animals and plants from inorganic matter, understanding this DNA code is synonymous to understanding life itself. So the instruction for life of a single cell, and also a life of a complex organisms like us, humans, which contains over 37 trillion cells, right? Each cell contains the same DNA, which is stored in a nucleus. When cells divide, as they do when new organisms is formed, or when new cells has to be generated, we see how this DNA is organized. The nucleus is opened up, and inside we discover these tightly packed strings that can be visible under a microscope. These are chromosomes. Chromosomes contains different genes. Each one of our cells contain 23 pairs of chromosomes. One of the chromosomes come from one half of the chromosomes come from our mother. The other half comes from our father. Yes. So this unique structure of chromosomes keeps DNA tightly wrapped around in a spool, wrapped around proteins like a spool, and these proteins are histones. So without such packaging into tightly wound chromosomes, DNA molecules would be extremely long to fit inside the cell. For example, if all of the DNA molecules inside a single human cell were to be unwound from their histones and to be placed as end-to-end as like a single string, that would stretch to reach to 6 feet. So the largest chromosome of all the chromosomes is chromosome 1, which contains about 8,000 genes. And the smallest chromosome is chromosome 21, which contains about 300 genes. So once the cell division is complete, the information on the cell that is stored in the DNA helps the new cell know what to do to continue its function that it will work on for the rest of its life, and preserve the DNA that it has been passed on from its parents and to pass on to the next cell. And in the meantime, the DNA that contains instruction for various functions in a cell must perform to grow and maintain life and function. Therefore, the DNA contains both the necessary ingredients like all the recipes that is required for the cell to function, and all the other instructions that is required for the DNA to be stored and accessed at the specific time. Together, all of the specific code of chromosomes that we are dealing with or that we are discussing with is called a genotype. The product of this code and how it interacts with the environment is called a phenotype. A phenotype can describe a single cell, a tissue, an organ, or even the whole organism. A phenotype of an organism could include height, size, weight, or activity. And phenotype of a cell could include its specific function, its size, its shape or morphology, and its composition. So DNA is made up of nucleotides at the basic level, at the base. It's a fundamental level. The DNA is made up of nucleotides. A nucleotide is made up of a sugar called deoxyribose and a nitrogen containing base and a phosphate group. So the nucleotides are joined to one another in a chain by covalent bond that forms between sugar of one nucleotide and the phosphate of the next, resulting in an alternative sugar-phosphate backbone. So each strand of a DNA molecule consists of a sequence of bases that is complementary to the bases in the opposing strand. So the pairing between these complementary bases is what gives rise to the characteristic double helix shape. So in this structure, adenine is bound to thymine and guanine is bound to cytosine. So to understand how DNA works and to read its sequence, a special project was aimed at decoding DNA that was launched in 1990. So this 13-year-long project was initiated in 1990 and is focused on determining the DNA sequence of the entire human genome. So scientists set out to determine the DNA sequence and the location of estimated 100,000 human genes at the time of the initiation of this project. Many at that time believed that once we had the sequence, we would quickly be able to determine where all the genes were. And this process of reading the DNA sequences and piecing them together laid the foundation of what we are doing today, laid the foundation for genomic study and the foundation for all the other omics study that is thriving today. Yeah. So, but simply reading the code and annotating it became a much longer process than sequencing the genome itself, than the Human Genome Project itself. Today, there are several different competing human gene databases with many thousands of differences among them. Although the number of protein coding genes has gradually converged and the number of other gene types has exploded. So the latest count has consistently has found over 22,000 genes long since the Human Genome Project was completed. So we have around 22,000 genes, protein coding genes, and that forms only 1 to 2% of content that we have in our whole genome. So in brief, the DNA is stored in the nucleus and it is organized into chromosomes and into specific regions. And some of these regions are easier to read and to access and to be evaluated for further functions. And these easy to read regions are called protein coding genes. And protein coding genes are the sequences, or the genes or sequences that is used to produce proteins. So how it is done? The central dogma of molecular biology describes how this is done. This is done involving a two-step process, transcription and translation, by which the information from the gene is converted first into RNA and then to protein. The information flows from genes to proteins through DNA being converted to RNA and RNA being translated to protein. So in each step of this transformation, we see how the DNA code is used. First, for transcribing the source code to a working repository of RNA, where some code is modified and some code is not used at all, and then it is released for producing a functional product called protein. So this process has facilitated several key proteins, one of which is facilitated by several key proteins. So one of which is called DNA polymerase. This DNA polymerase is the one which attaches to the DNA as it unzips to replicate the DNA to form single strand and and then it adds complementary bases to the single strand and creating a second workable copy. This copy DNA code can in principle be used in other cells also, but yeah, it is can be used in other cells also, but it is also important to make sure such a replication process goes without any mistake, so that the cell doesn't end up with a different set of copied DNA than to the original set. And the cell also has a lot of mechanisms and other products and instrumentation in protein complexes that are designed to prevent this error or in error correction mechanisms also. So each gene, as we saw, is a recipe for certain protein. So the gene sequences encodes instruction from which a protein can be made, and the DNA sequences encodes the instruction from which a protein can be made. So proteins are the products of amino acids, which are encoded by the three sequences or trinucleotide sequences that are called codons that are stored in the DNA. So the DNA sequences they are read by the jump of three. First, three sequences are read and then converted into one amino acid, and then goes the next three, and then goes the next three. So a codon is a sequence of three nucleotides that corresponds to specific amino acid or a stop signal during a protein synthesis process. Once a right place on DNA is copied over, it is prepared for translation by forming a messenger RNA that is exported from the nucleus. So the translation happens in the cytosol, not in the nucleus. So in humans, we saw genes are very important and they make up protein. And in humans, genes vary in sizes from few hundred DNA bases to more than 2 million DNA bases or 2 million nucleotides. The keratin-associated protein, for example, is 447 nucleotide long, but whereas CNTNAP2 gene has something like more than 2 million nucleotides. So the number of genes also varies between various organisms. For example, the influenza virus has only 11 genes, while the grape genome has over 30,000 genes. So the information is not uniform across the different organisms. Neither the information about coding proteins is uniform across the genome itself. So the genes are not just sequences of nucleotides. In fact, they have regulatory sequences at both the ends of the gene, that is upstream and also downstream. This regulatory sequences controls the level of transcription of each gene. So inside the gene sequence itself, there are protein coding regions called codons or exons and non-coding regions called introns. The cell's machinery perform a process called splicing that will leave out or that will exclude the introns and then include only the exons before the gene is translated into proteins. And the extraordinary length of some of the genes has enabled the presence of introns, and this presence of introns and multiple introns provides a mechanism for generation of transcriptional diversity through alternative combinations of different exons at different levels. So the translation in the cell is done by ribosome, which can read the mRNA sequence and assemble the protein, and this protein can then fold into a functional form or functional structure. So can you see my screen? Yeah, I think yes, now it is working. Yeah. So when we want to understand the function of a particular protein, we typically combine the information about its chromosome position, about the coding sequences that it has, about the regulatory region, about the structure as well as functional features of the protein, and about all these information which are understood through thorough experimental validation and or its comparison to proteins from the same family or proteins that has the same structure. So now we understood the context of what is DNA, how it is organized, and how it is used, and how it is translated to form protein, which are the machinery that defines the characteristics or that defines the phenotype of the cell or that defines the functions of the cell. So let's discuss how to analyze this DNA code. Let's look at the sequence alignment and identification of variant that have functional consequences on the phenotype of the biological system currently under study. So let's go back to a little bit of a history. So the year 2020 marks a 70-year mark since the discovery of structure and function of DNA molecules. It took almost 50 years from the time to sequence the first human genome, but soon after, the technology evolved and exploded rapidly. And by 2050, we have the 1000 Genome Project, which brought a significant number of whole genome sequences to the research community. And in 2018, we have over 100,000 whole genomes that were sequenced, and today this is an ever-increasing number of different omics data sets that are available that will describe cellular and subcellular process in a detail that cannot be imagined something like 50 years ago. So the omics, the multiple omics that you just heard, aims at a collective characterization and quantification of pools of biological molecules that translates into structure, function, and dynamics of these organisms or multiple organisms. The omic technologies are used to explore the roles, relationships, and actions of various types of molecules that make up the cells of an organism. So many types of omics data that can be generated using high-throughput sequencing or next-gen sequencing can show detailed information about genomic variants, epigenomic regulation, phenomic data types, and the different transcriptomic states of the cells that we have in our study. The suffix "om," one used to define various fields, actually originated from in the 20th century. In the 20th century, when genome was first coined in Germany in the beginning of in the 20th century. The word genomics refers to the totality of the genomes, or the study of all the genes or genomes that is present in the cell. A genome is an organism's complete set of DNA, including all of its genes, as I mentioned, in contrast to genetics, which specifically refers to study of individual genes and their role in inheritance and their characteristics that defined from this inheritance. Genomics aims at a collective characterization and quantification of all the organism's genes, their interrelations, and influence on the organisms. As a result of such effort, we can now detect differences which can be harmless or differences that will lead to serious issue when a gene is produced. So just like any code, we can expect the DNA code to contain mistakes. So these mistakes occur as a result of copying the DNA source too many times, as a result of various stresses that can interfere with this copying mechanism that is inside the cell. So to understand how genomics is used, we need to understand the analysis approaches to DNA code, specifically how we can identify the changes or how we can identify the mutations that characterize them, and to interpret the significance of these changes that we naturally observe, or that we observe when we sequence the DNA. So mutation can actually happen in several different ways. So one form is substitution. A substitution mutation is something that can cause several consequences to DNA code, but it implies a minimal change that only leads to a single nucleotide change. So when one nucleotide is substituted with the other nucleotide, it's a straightforward substitution. And insertion, however, changes the number of DNA bases in a gene by adding a piece of DNA. As a result, the protein that is made by the gene may not function properly because the reading sequences have completely changed due to the insertion of this one or multiple DNA bases. A deletion mutation, however, changes the number by removing one or certain pieces of DNA. Small deletions may remove one or few bases within a gene, while larger deletions can involve removing an entire gene or an entire exon of several neighboring genes. deleted DNA sequence may alter the function completely alter the function of the resulting protein, and that is going to interfere with the cell's processes. So a duplication mutation or duplication event consists of a piece of DNA that is abnormally copied one or more times. This type of mutation also alters the function of the resulting proteins, and which will also interfere with the cell's machinery. So a specific type of variation can lead to harmful results. For example, a change in function is harmful, and this is called mutation. Or in the other way, when the changes in these variation is not harmful, then that will not be that is not a that's a neutral mutation, or that is no mutation. That is a variation. So for example, in this case, variation causes the eye color to change, while mutation causes some really horrible deformities and functional losses. So when these genomic variants are discussed, both mutation and both the term mutation and polymorphisms are used interchangeably, but by convention, in human genetic research, any genomic variant with the population frequency of less than 1% is termed as mutation, while any genomic variant with the frequency of greater than 1% is termed as polymorphism. So this distinction based on the population frequency of the variant rather than the type of the variant is based on the variant or the type rather than the type of variant or its possible functional impact. However, as a rule, rare mutations that are rare, that is even less than 1% observations, tend to have functional impact that deviates from the wild type, yield to higher signal to noise ratio for detection of a genetic influence of a trait, or a rare mutation that will have functional impact can have very drastic effects in the influence of the trait. In contrast, polymorphism that is observed tend to result in less functionally deviant genomic variants. It might not cause severe deformities or severe functional impact of the cells or of the organisms in study. So, however, it is also worthwhile to think, or it is also worthwhile to keep in mind that these rules, there are exceptions to these rules. It is also observed that polymorphisms can be, some of the specific polymorphisms can involve loss of function, and some of the simple variations in gene mutation can lead to no loss in function at all. So, a single nucleotide mutations can be of several kinds. So this is a single nucleotide mutations. Substitution mutations can be of several kinds. One of this kind is missense mutation, which changes only one DNA base pair, and that results in a substitution of one amino acid for the other in the protein made by the gene. A nonsense mutation, however, changes one DNA base pair that prematurely causes the cell to stop building the protein, or it introduces a stop codon in the DNA sequence that prematurely stops the length of the protein that is being synthesized. And this type of mutation results in a shortened protein which will not, or which might not, function properly at all. There is another type of mutation which is a frame shift mutation, where an addition or a deletion of DNA bases completely changes the frame of the gene's reading. As I had introduced you before, the codons are read 3 bases once. So when you delete or insert one or few bases, this reading frame changes, and by changing this reading frame shift mutation occurs, and this will result in a protein that is completely different from the original or completely different from the wild type protein, and this could usually result in a non-functional protein. So to summarize these different genomic variations. Genomic variations can be of several different types. So when analyzing the genomic variants or genomic data, we are looking to find and characterize these changes in the DNA code and associate such changes that we are observing to the phenotype or to the different cells characteristics. Here are some of the acronyms and some of the words and jargons that you have to remember about genomic variations: SNPs and SNVs. So SNV is a single nucleotide variant, where a single nucleotide change that may or may not be present in the population. Basically, this means any change from the reference genome that we find in a given DNA sequence above the level of technical error. So SNP is on the other hand, is a single nucleotide polymorphism, where a nucleotide is known to be altered in a proportion of population and typically considered to be prevalent in approximately more than 1% of the population. So this is a single nucleotide polymorphism is a mechanism that causes blue eyes and green eyes and different eye colors in the in different human races. So another form of acronym that you should remember is copy number variation, where the same gene is encoded, or same gene is being copied multiple times and is present in different number of copies across different cell types or across different conditions of the cells that is under study. So such changes as well as the characteristics of these different variants are commonly referred to as genotype of the cell or genotype of the organisms. So this is what we are studying by understanding or by sequencing the DNA sequence, and by trying to analyze the DNA sequence, we are trying to understand the genotype of the organisms, or we are trying to understand the genotype of the cells. So the bioinformatics technology that will allow you to generate the digital information from these biological samples is called next-gen sequencing or high-throughput sequencing. This technology was first developed during the Human Genome Project, as discussed ago, and has evolved tremendously since then. So over the years, many different sequencing techniques were introduced, and they were developed and perfected to study whole genome sequences of multiple organisms. So sequencing platforms use variety of read lengths. So read lengths is the length of the DNA sequence that can be read at once, and this varies by different sequencing platforms. But in general, they can be divided into short read sequencers and long read sequencers. So the benefit of the short read sequences isn't that because they are short, but they can be extremely inexpensive, and they can be generated in a massively parallel manner with billions and billions of reads being generated in a single run. So the cost, when the cost needs to be kept down, and when you have to sequence multiple different samples, and you have to perform it a lot of times, and the read count has to be really high, and then short read technologies makes most sense. While long reads, on the other hand, can resolve regions of genome that are inaccessible or that are inherently not possible to assemble due to the length of the short reads, because these regions can involve repeat sequences, and those repeat sequences may interrupt our read mapping process. So in that case, long read sequences have advantages over short read sequences while dealing specifically with those regions which have repeat sequences. And latest advancements and recent advancements made by Oxford Nanopore Technologies, for example, under right conditions, under perfect conditions, under perfect high quality DNA, can even produce reads up to 1 million bases. That is huge. So this technology allows researchers to explore the genetic variations like never before, down to a single nucleotide resolution. Whether you need to identify specific variants that from a focused set of regions, or to look more broadly about potential different variations or causative variations, or to look for differences or variations in the number of variations in the number of times a gene is being copied. So these are all different explorations that one can perform. So based on this, whole exome sequencing or whole genome sequencing can both offer effective solutions based on your question to what to look for and how to design your data. Whole exome sequencing or whole genome sequencing, as I mentioned, can be both effective. So whole exome sequencing is a targeted method to sequence all protein coding regions in the genome. It offers reliable and sensitive detections of coding variants, and it is fast and it is cost effective, as it sequences only a fraction of what the whole genome sequencing is going to perform. Whole genome sequencing, on the other hand, provides a comprehensive view of the genome, including coding, non-coding, mitochondrial DNA, and everything that is present in the cell. So it is reliable and sensitive as well, but includes the process is complicated, and it also it can provide you with the necessary information about detection of different variant types that is under study or that is under question. And these variant types or these variations and mutations, these variations are not limited to coding regions alone, because we are sequencing the whole genome. So now let's understand about how this process is being employed and how this is being done in a very short intro. So the NGS technology that captures sequences the DNA bases actually captures and generates this digital data using several steps. Here, the whole genomic DNA is isolated, and then it is sheared, and it is shattered and sheared and placed in a flow cell. So inside the flow cell, the DNA fragments are amplified using bridge PCR amplification, and then the flow cell is inserted into the sequencer. The sequencer machine uses image analysis to capture each and every nucleotide in this flow cell fragments by analyzing the visual patterns and converting them into sequence of letters, that is TCGA, that are the nucleotides that forms the bases. Eventually, the sequencer will produce reads typically of 30 to 300 base pairs, base pairs in length. So this sequencing data that is emerging or that is coming out of the sequencer is stored in several different file formats. So for example, a FASTQ file format will provide us reads with both, will provide us the reads from the sequencer, and it is text-based file format. This stores the nucleotide sequences of reads and the quality scores for each letter that is sequenced. The FASTA format, on the other hand, stores the representing DNA and protein sequences, and this is typically reserved for the reference file of the genome or reference file for the genome to which the reads that are in FASTQ files are matched to. Then we have the genome transfer format, which contains the genome annotation. For example, a genome transfer format, GTF file, will store the start and end of each genomic element like exon, like isoform, or like gene, and other sequences, other elements that you find in the whole genome sequence. So, can somebody mute their microphone, please? Yeah. So in short, a FASTA file contains a genomic sequence itself with a line that's starting as with the line that starts with the greater than symbol, which is a header that describes a sequence, and this is and describes the sequence. For example, this header describes a species from which the sequence is for which the DNA sequence belongs to. In this case, it's from a virus, and in this case, it's from a Frugiperda fungus. So a FASTQ file, on the other hand, is also a text-based format, but it stored the sequence, and it also stores the corresponding quality scores of each and every letter that is being sequenced. So both the sequence letter quality scores of each RE are encoded in a single ASCII character. So a FASTQ file normally includes information about the name of the read, its length, the raw sequence letters, and the quality values for each sequence for each nucleotide. Depending upon the sequencing method that is being employed, FASTQ files can be quite large, sometimes over several gigabytes in size per file. So to analyze such raw data, many different tools for genomic data analysis are used. These analysis tools include alignment of reads and full sequences, analysis of relationship between sequence and their evolutionary divergence, and measurements of gene expression, and relationship between sequences and structures, as well as relationship between somatic and germline variant calling methods, and multiple different techniques that will characterize somatic and germline variants. So the platform, the tBioInfo platform, the tBioInfo is a big data platform that can be used to perform all of these analysis and many other using an intuitively user-friendly interface. So the sequence alignment that I mentioned ago can refer to two types of alignment. The sequence alignment to a reference, in which case it is called mapping. So where you have the FASTQ files, and these files are from the sample, and you wish to align these files or reads to its best position on the genome to find whether the sequence in the read or multiple reads reveal variation from the reference which are in FASTA format. So in other type of alignment, which is multiple sequence alignment, this is what you do when you have multiple consensus sequences that you would like to compare each one of these sequences for their differences or for their similarities, and develop an understanding between them in terms of how these sequences or portions of these sequences are conserved or portions of the sequences are varied. So in many cases, these multiple sequence alignments are used to find structurally important variation, and it can also be structurally important variation in both nucleotides and also in protein sequences. So when we have the FASTQ files ready from the sequencer, and when they are ready to be analyzed, there are several important steps to be considered for performing the analysis. These steps include pre-processing, mapping, variant calling, and annotations. So for each task, you can connect analysis jobs to a pipeline. Each one of these various inputs, a pipeline will have various inputs, and each of these algorithms in this pipeline will have multiple outputs, and a pipeline is defined by joining several algorithms together, from where the output of one algorithm straight away or directly feeds to the input of the other algorithm. And in tBioInfo platform, the outputs at every step is presented to you and is given to you, and you can analyze or you can visualize where the information is changed and how the information content is built upon, and how the information content is maximized at each step of the at each analysis step of this pipeline. So however, to carry out a genome-wide analysis study, for example, our researchers use two groups of participants, let's say people with the database, people with diseases being studied, or people with without diseases. Sometimes this genome-wide association studies can also be performed to identify certain variations of disease or conditions that we have in hand. So how is this performed is when the researchers obtained DNA from each participant and sequenced their whole genome, and if a certain genetic variations are found to be significantly more frequent in people with the disease compared to people without the disease, then these variations are said to be associated with the disease. So identifying these associated genetic variations can serve as a powerful pointers to understanding those regions of human genome where the disease causing problem resides. So the methods that I just mentioned before, downstream analysis or visualization and somatic mutations, can help us understand and can help us relate these genetic variations to these different regions which might be involved in causing diseases or causing problems. So yeah, so in this case, it's easy to understand, or it's easy to observe that chromosome 14 has a higher mutational rate that could be involved in the disease causing state. So downstream analysis will actually identify mutations, SNP variations, and these mutations and SNP variations can be presented in multiple different ways depending upon the purpose of analysis. And in this case, it can be presented in a tabular format, or it can be presented in a simple graphical formats, depending upon depending upon the kind of report that you are generating based on the analysis that you just perform. So this program is actually designed to help you learn more about these analysis methods and their applications in various project examples. So this genomics program will include an expanded overview of key concepts, principles, and examples of genomic data analysis. Now I want to invite Dr. Harpreet to present you with a couple of demonstrations of such analysis which can be performed in TBioInfo Big Data platform. So over to you, Dr. Harpreet, and then we will come back for the for how to briefly talk about how such genomic studies can be used in